Hey, hi everyone. Thank you for joining us. And it's wonderful to see how many people have booked onto today's webinar. I'm Jane Ackerman and I'm Programme Manager for the IUCN UK Peatland Programme. I'll start with some housekeeping, then a very brief introduction to the Peatland Programme and our biodiversity advocacy work before I hand over to the first of our brilliant lineup of speakers. Today's webinar is being recorded, but only our speakers will appear in that recording. So please use the Q&A function in Zoom if you have a question for one of our speakers. We'll be holding a Q&A session at the end of all the talks where I'll put as many of your questions to the speakers as time allows. You can also upvote your favourite questions in the Q&A feed. You're welcome to use the Zoom chat function during the webinar to make comments as well, but don't use this to ask questions as they may be missed. Please make my life as easy as you can in chairing this session. So I'd like to start with a very brief introduction to the Peatland Programme for those of you who may be unfamiliar with our work or perhaps aren't aware of our wider efforts to celebrate peatland biodiversity this year. Founded in 2009, we're an umbrella organisation which represents a really broad range of partners, including government and devolved agencies, non-governmental organisations, researchers and regional peatland partnerships, all working together for the restoration, protection and sustainable management of UK peatlands. We provide specialist advice, help to inform policy, produce publications and other resources to educate and inform people about peatlands, and we deliver the Peatland Code, a voluntary carbon finance standard for UK peatlands. Our vision is that peatland ecosystems are valued based on the many ecosystem services they provide. With around 80% of UK peatlands in a degraded state, largely due to drainage for agriculture, forestry or peat extraction, there's still a long way to go, but there's also a lot to celebrate, with a growing number of peatland partnerships dedicated to restoring and protecting these wonderful habitats by working together across multiple organisations. There is increasing understanding of the importance of healthy peatlands as incredible carbon stores, and restoring degraded peatlands is a nature-based solution to the climate crisis. However, we're currently facing another connected crisis, that of catastrophic biodiversity loss, and peatlands are sadly no exception. A big part of the Peatland Programme's current work is to educate people about the unique biodiversity that peatlands are home to and why that biodiversity matters. We also want to celebrate the many projects engaging people with peatland species and the restoration projects bringing these species back. Our March newsletter was biodiversity themed, so if you missed that, you can find it on our website, as well as the first two of our species showcases on the sphagnum bog mosses and a very cool group of microorganisms called testate amoebae. If you're interested in sharing your work as part of one of our species showcases on our website and social media, then please do get in touch. Well, that's enough from me, and I'm really excited to hand over to the first of our speakers today, and that's Brian Eversham, who is CEO of the Wildlife Trust for Bedfordshire, Cambridgeshire and Northamptonshire. Brian's a brilliant photographer and a fountain of knowledge on all things peatlands, and he's going to introduce us to peatland biodiversity and the importance of species identification skills in biodiversity monitoring.
So first slide, loads of different species and loads of different groups. And I should say, from what I've seen of the audience today, it's great there's so many of you here, but it does make me feel that everything I talk about, there'll be someone in the room who knows more than I do. But my job in the next 15 minutes is to give a very brief overview of UK peatlands, their geography, their biodiversity, why species matter, how you choose taxonomic groups to work on, how you teach species identification, and finally just touch briefly on why basic taxonomy is important, even if you're not doing basic taxonomy, if you're using new and advanced techniques. So to start with peatlands, they are spread across the country. They look like a predominantly upland and northwestern group, but if we look at the range of peatlands, we can run through and see there are important and very biodiverse peatlands in all sorts of places. So most of the upland peat is blanket bog. Some of the lower altitude peats are raised bogs, the largest ones in South Yorkshire, but also some in Somerset Levels and a few other places. And scattered through the lowlands, there are valley mires. And then there's the fen basin, which is a slightly odd place, which I'll talk about in slightly more detail. As context for looking at peatland biodiversity, it's worth noting that in Britain as a whole, most biodiversity shows a very strong southeast to northwest gradient. So the lowland, warm, sunny south has more species in almost all groups of plants and animals than the cool, wet uplands of the north and west. The exceptions to that, which become quite important when looking at peatlands, mosses, liverworts, ferns, horsetails, sedges, and craneflies all have more species in the north and west than in the southeast. Almost every other group of plants and animals goes the other way. Starting up top, blanket bogs, generally flat, often quite deep peat, and largely rainwater fed and sphagnum dominated. Raised bogs, when intact, would have a domed profile, often very deep peat, quite often 10, 12 meters deep. Almost all our large raised bogs have been seriously damaged by peat cutting, but they are still wet and they still support sphagnum and a range of other things. The fens are rather curious in that here we're looking at peatlands, which is much more varied in topography and chemistry, often with groundwater influences, often more calcareous, and with its own set of important species. And valley mires often occur as relatively small sites embedded in mosaic, including heathlands and successing through to woodlands. And the New Forest has some fabulous valley mires. So purpose today really is, why do we want to know about species in peatlands? And I reckon it's mostly about evaluating sites, monitoring sites, looking at the health of sites. And the warning today is that the most obvious groups of plants and animals to look at and the ones that are most popular aren't always the ones with the best ecological signals. So I'll start with the peatlands I know best. This is part of Hatfield Moor, one of the two large lowland raised mires in South Yorkshire. Hatfield is the second largest raised mire in Britain, Thornmoor's the largest. Both sites, like most raised mires, have been cut over and drained for 150 years or so, but they still have an enormous biodiversity. We're looking at about 2 square metres of a rather drained part of Hatfield, and the starting point is to say, what can you see in the picture? And in those 2 square metres, there's lots of bracken, the common fern, there's a bit of birch, cross-leaved heath, haresdale cotton grass, and a little bit of heather, so 4 common flowering plants. On your hands and knees, you'd find 3 common cladonia lichens, 5 mosses, one of which is a little bit less common. The whole area is used by a small number of bird species on this site, including nightjar and woodlark, both of which are pretty scarce. It's visited by 7 common mammals, 2 common reptiles. Not a lot there to evaluate the site with. And yet, next to this puddle, one pitfall trap in 1 year collected 330 species of insect. The most abundant insect in the trap was a fungus gnat new to Britain. There were 5 species in that pitfall that are unique to this pair of peatlands and the Humbered Levels. One of those is what's colloquially the Thornmoor's ground beetle, Bembidia numerale, only 3 millimeters long, and its modern British range is confined to that one very small area. There's a Bronze Age fossil remains from the Somerset Levels, so it has been more widespread on lowland peatlands, but these days confined just to the one area. And if we look at a set of peatlands which have been thoroughly audited for the biodiversity, the fens, we know that the fens support over 13,000 species of plants and animals. But looking at the ones that are unique to the fens, rather shockingly, 3 flowering plants, 2 species, 1 subspecies, a mollusk, a couple of spiders, and lots of insects. And this is saying a lot about the nature of peatlands. There's a huge invertebrate biodiversity, which is not always as well known as it ought to be. In the fens, almost every group of invertebrates you look at have some special species confined to that part of the country and associated with a range of habitats supported by fens. Almost all peatlands have at their base sphagnum bog moss. It's featured in most of Jane's slides to start with. I'm sure will crop up a lot more during the talk. And you probably don't need me to say sphagnum is a uniquely important and charismatic plant. Its structure with large open spaces between the cells means that it can absorb many times its own dry weight in water. The chemistry of sphagnum means that it can absorb metal ions, pump out hydrogen ions, leaving acid, low nutrient water, which supports that very unique range of communities associated with peatlands and preserves the plant and animal materials themselves as well, hence laying down peat and becoming the world's biggest and most important carbon store. And sphagnum isn't a single species, it's a wide range of species, 40 odd in Britain, differently coloured, differently structured, with very different ecologies. You've got sphagnum that grow in slightly damp woodland, through to ones which are completely submerged, some which form extensive lawns, some which colonise damaged or burnt over sites, and several of which form the major hummocks that give you the wonderful microtopography of healthy, intact peatlands. So sphagnum is one of those groups where knowing the species changes your perception of what you see in the habitat. Surprisingly enough, testate amoebae have cropped up already. I find them a lovely group to work on. I love the microfauna associated with sphagnum. Take a drop of water out of a handful of fresh sphagnum and you will find rotivers and tardigrades, gastrotrichs, and dozens of testate amoebae. They're very well known in peatland studies as part of the paleoarchive, in the same way as pollen analysis of insect fragments can be used, looking through very long-term changes in peatland. I'd suggest they're also equally valuable looking at the structure and function of modern live peatlands, and we should think of testate amoebae as wildlife, and we should go out and record them as we record other wildlife. I thought I'd take a pause now just to discuss why species identification rather than broader classifications really is important if we want to understand peatlands. And I've taken two common ants found on peat bogs of all sorts. Both of them dark brown, long-legged, quite alert looking, quite active. Does it matter if we call them one species or two or three or four? Well, the idea that naming is important goes back at least 2,500 years. And at the very least, I want to be able to say we're looking here at Formica fusca and Formica lemani. Why does it matter? Well, look at their geographic distribution. If you've got a pair of closely related species, one northern and one predominantly southern, then something is going on that's different. You will find that these species have different ecologies, different behaviours, different uses of habitats. And so generalizing between those two is going to lose important ecological signals. But it gets more important when we look at one of their close relatives. Formica picea is a species which is incredibly rare in Britain and across much of Europe, is confined to lowland valley mires, and it's verging on extinct in Britain. If you regarded long-legged black ants as one thing, you would miss out that vital ecological signal. And this is the most peat boggy of all the ant species we have. One of my other favourite groups, the ground beetles, the largest genus in Britain and across most of the North Temperate in the ground beetles, Bembidian. They're tiny animals, the biggest one's 6 millimeters long, and the Venn diagram shows how they divide up different wetlands. There's a whole set of species associated with river shingle. Most of the standing freshwater species do occur in fens. There's a handful of species specific to lowland raised bogs and other very acidic habitats. And three of the closest related species really show why naming species matters. Quadrimaculatum is a common species in gardens, on arable land, and in wetlands. Quadripustulatum is only found in highly calcareous wetlands. Humerale is found on sphagnum bogs only. So unless you know which species you've got, you're losing important ecological information. In my wildlife trust, when we're planning training workshops, this is one of the frameworks we use. We take the full range of British plants and animals, classify them as to how useful they are as broad indicators of habitat quality and diversity and that ecological signal gain. So on the right-hand side, you've got a matrix of broad habitat types and scores between 0 and 3. If you score 3, you've got lots of species tied to that habitat and good indicators. If you score 1 or 0, you're not much use in that habitat. And by doing this, we can actually work out which groups it would be worth teaching people. And I have to say, with running training workshops for the last 30 years, the more difficult and challenging you make a workshop sound, the more people will turn up for it and the more diverse audience you will get. If I run an Introducing Wildflowers workshop, I might get half a dozen or a dozen participants, and if I can be ageist and sexist, they will tend to be comfortably off, middle class, and retired. If I run identifying indicators of calcareous grassland or heathland lichens or sphagnum species of raised bogs, I have a much larger audience and a mixed audience, and young people really want to learn. It's a gap in university teaching which young graduates are keen to put right. And if we offer detailed local training, people will sign up and you will get a range of volunteers and a range of professionals who want to know and do more. And the resources behind training have improved hugely over the last 10 or 20 years. There are groups now like freshwater algae, plant galls, sphagnums, where there are really good up-to-date guides. And there are some groups where we've not had a good up-to-date guide for decades or centuries. About 5 years ago, 2 new books on bees came out, first bee guides for a century. We've got really good guides to micromoths. You can now identify British dandelions to species, all 250 of them. So the literature and online resources enable us to do a better job teaching the precision of biodiversity that we need. And the other point I'd stress is that if you know where you're monitoring and what you're monitoring, you can simplify the life a great deal more. If I tried teaching somebody British lichens, well, with 1,700 species, it's going to be years or decades worth of work. Even in bedcams and Northants, the relatively flat species poor bit of central England, we've still got 500 species to learn. Narrow it down to lichens growing on bogs, fens and moors, and you're down to just 3 or 4 genera. And in the most complex genus of all, the cladonias, you go down from 60 odd species nationally to only 15 or 20 in lowland England. So I could teach you all to identify cladonias to species in one day, rather than the decades you might need to learn the rest of the lichens. So focus like that, and you can give people skills. The other thing to say is that the world of the internet has suddenly brought specialists in touch with learners. So if you're beginning with the cladonias on Twitter or on Facebook, you can find people who are eager and happy to help you identify what you're finding. So all of us can be part of a teaching and learning network. And a final thought, most training workshops tend to be based on the tradition of using identification keys, an analytical approach. Life isn't always like that, though. Everybody in the room can tell me instantly that you're looking at a dog and a cat there and which is which. If I were to write a key to identify dogs and cats to separate every sort of dog from every sort of cat, the 2 features which you can just see in the photo, which would separate dogs from cats, would be vertical slit pupils in cats, round pupils in dogs, retractable claws in cats, and sticking out claws in dogs. Tiny features that you never notice and you never use to recognize dogs and cats. Likewise with most wildlife, there are shortcuts which you can give to trainees which will allow them to recognize the most important species without faffing around for hours going through identification keys, though I'm sure keys are good both for learning a group and for the soul. A very brief deviation from peat bogs to one bit of primary taxonomy I've been involved in recent years, just to make the point that whatever techniques you're using, you may want some proper traditional taxonomy. Depending on which book you look at, there are anything from 2 to 62 elm species in Britain. I'm of the 62 species tendency. One reason I believe in them is that serious research has demonstrated that the insects, the plant galls and leaf mines can recognize the different microspecies of elm. If it's good enough for the insects, it's good enough, I think, for the botanists. And a couple of years ago, a global phylogeny of elms demonstrated that the little purple block, which some people would call one species in Britain, actually has as much genetic variation between it as well-established species elsewhere. I put this story in, though, not just because elms are fascinating and worth talking about, but to make the point that unless you have the traditional taxonomy and people who can point you at the full range of species, DNA work is pretty futile. You'd never sample those microspecies of elm without a traditional taxonomist to guide you. So although DNA work can provide us with lots of answers and lots of useful probes, we do need it to be based on pretty traditional taxonomics as well. And I think that's about all I wanted to say. So if you have any questions, please type them into the Q&A and we'll answer them or try to answer them toward the end of the meeting. And I'll stop sharing at that point.
That's fabulous. Thank you, Brian. I was an entomologist in a former life, so that very much appealed to me. Emphasize the importance of all those wonderful invertebrates on peatlands. So many things to explore and discover. That's brilliant. So next up, we have Jenna Lawson. Jenna is the Ecological Network Coordinator at the UK Centre for Ecology and Hydrology. And she's going to talk to us about her work in monitoring biodiversity using some very cool technological approaches. So I will hand over to Jenna now. And as always, any questions for any of our speakers, pop them in the Q&A and we'll get round to them at the end of the session.
Brilliant, fantastic. Hi, everyone. So my name's Jenna. I work for the UK Centre for Ecology and Hydrology. And I think I'm going to upset Brian a little bit by moving from traditional taxonomy over to the world of tech. So the aim of technology isn't to replace the traditional methods. I think the way we see it is more of a complementary approach. So today I'm just going to discuss some of the key forms of technology that are out there, not necessarily relating them to peatland in any way, but hopefully from the examples that I share, you might be able to see how they relate to peatland environments. So a little bit about why technology is important. So I think at the moment, there's a lot of, we know that the collection and availability of good quality data, as Brian talked about, it's essential for better understanding and addressing those key information gaps that we have, which then lead on to improved decision making. And I think there's a famous phrase that we cannot protect what we don't know. And currently, it has been said in many journals recently that we do lack the evidence to effectively protect and manage many vulnerable species. And technology is one area that I think can be used to actually increase that evidence base to help us make better decisions. And so I always like to include this slide because when I think about what gold standard data is, I see technology as being able to help with providing gold standard data. So I think technology not sort of gets rid of invasiveness of the work, but it can reduce the invasiveness of the work that we do. It can reduce the bias in taxonomy, the geographic bias. It can reduce bias in human identification. It can increase the spatial and temporal scale of what we do, which is really important, especially if we want to make conservation decisions. It provides us with that high resolution data, and it can provide us with data that's standardized and verifiable, which is becoming increasingly important. And so some of the technology that's available, I'm sure a lot of you will be aware of most of this technology. So AI is the big one on everyone's lips at the moment, whether for good or for bad. Acoustic monitoring. I'm going to talk to you today about the AMY system, which is a new piece of tech that's come out recently from UKCEH. Drones and robotics are becoming increasingly used in ecology, camera traps, which have been around for a long time, biologgers, eDNA, which has been around for a very long time in the microorganism world, but is making its way into the macro world at the moment. And the use of satellite data is becoming increasingly used. So today I'm going to talk, AI is going to be a common theme throughout everything that I talk about, but I'm going to talk to you about acoustic monitoring, the AMY system, and a little bit of work on drones and robotics at the end. So I'll start off with acoustics, which is my expertise. It's where I did my PhD. And when we're looking at acoustics, we're basically putting up acoustic monitoring equipment in different environments, for example, in a peatland. And then when we collect in all these thousands or hundreds and thousands of hours of recording, we have a few ways that we can analyze the data. And we can go through that data manually, but that would take us a very long time. So often we're employing AI measures or soundscape measures. So the first one of these would be species-specific measures. And so what we can do is, everything I talk about today, I'm going to use examples just to show you how it can be put into practice in the field. So these maps here, these are maps of Costa Rica, so very far from UK peatlands, but just to show you as an example. The map on the right hand and the left hand side is a habitat map, and you can see all the different colors of different habitats. But basically, I had 400 audio recorders out over this area, and all the points in pink are where I didn't find my species of interest, and all the points in white are where where I did find my species of interest. And from this, I was able to tell a lot about the species distribution. And I was able to produce a habitat suitability map, the map that you can see on the right-hand side. And so from having these audio recorders up, you can tell whether the animal is in that location or not in that location if you don't hear it, with some detectability issues. So that's how we could use acoustics to look at species-specific information. And you can do this, imagine, this was for the spider monkey, but you could do this for any species that makes a sound. I'm not going to go into this in detail, but I'll go into the detail about the limitations of technology towards the end. But if anyone's interested in any more depth, there are some sort of limitations that I've included here for the different types of acoustic analysis. And so another way that we can analyze acoustics, it takes a very long time to produce species classifiers for individual species, to identify species to individuals. And sometimes we don't have that time and expertise. Sometimes we want just more broad measures of what's going on in an ecosystem. And this is where soundscape analysis comes in. So what we can essentially do with all these acoustic recordings that we collect in is that we can measure the acoustic energy, the amplitude, the frequency. We can measure all of these metrics from those sounds. And we can measure these using what's called acoustic indices. And essentially what these acoustic indices are doing is building up a broad picture of what's going on in that 1 minute or 1 hour sound recording. And just to give you an example of what happens, each of these black boxes here are a dawn chorus in a different ecosystem. So the first black box is an old growth forest, then we have a secondary forest, and then we have two different forms of plantation, a teak plantation and a palm plantation. And essentially, the more colors and lines in these black boxes, the more sounds, biotic sounds that are in this ecosystem. And so you can see the amount of sounds in an old growth forest, the first one, for example, versus a palm plantation. So you can see that visually just using these graphs, but we do have a lot of numbers and metrics that we can put onto these to show you the level of sounds within these ecosystems. Okay, I'll just skip over that one. So that's a very brief whiz through acoustics and how you can look at what species are in an ecosystem and how you can look at generally what biotic diversity there is in an ecosystem. So moving on to the AMI system, this is a very new system. It's developed in-house here at UKCEH, and the idea is to automate moth monitoring. So AMI stands for Automated Monitoring of Insects, and the AMI Plus is there because we're adding on acoustics to this system right now. So it's AMI Plus, which is standing for all of the biodiversity that can be recorded with sounds. And this picture here is a picture of the AMI that we were setting up this year out in Costa Rica. And so the AMI system, if you look at the images on the right-hand side, it's made up of a whiteboard which the nighttime flying insects land on, and they're attracted to that whiteboard with a LEPI-LED, a UV light. So it's not just UV; it's got blue, white, and green lights in it. And so they're attracted to the board. They land on the board, and then there's a motion-activated camera that takes pictures every time it detects motion. And then all of these images are then stored onto an SSD card in the back where all the other electronics are, like the Raspberry Pi, the Witty Pi, everything else that's controlling the system. And then we essentially go every month that we pick up the SD card and then we download those images. And you can customize the sampling schedules for this, so you can get it to turn on sort of 1 hour a night, 10 hours a night, after dusk, before dawn. And we have an AI, so I'll move on to that in a second, and it's powered by either battery and solar panels, or it can also be plugged into mains. So essentially, if this is being powered by battery and solar, you can leave the system out for months at a time. And even though we go and collect these SD cards every month, you can actually leave the SD card out for almost a year, just collecting images. And so this is a very brief video of the AMI system in action in Costa Rica. I've only got 15 minutes, so I won't show the entire video. And then just to give you a bit of information about how we would analyze this, again, using AI. So we have a species classification as you would classify acoustics. This is a classification for images. So it's a 3-stage classifier. It starts with object identification. So if you imagine the whiteboard with all the different images on the back, and what it does is it first identifies where it sees an object, and it draws a box around the objects. And then it's been trained in the second level of classification to identify a moth. Is it a moth or is it not a moth? Because at the moment we only have the species classifiers for moths. And so we're not throwing away all the other data right now. We're keeping that until we can develop species classifiers for all the other species, which will take some time. And then once it pulls out all these moth images, it then there's a third stage of the classification which attempts to classify it to species level for that moth. And so this is just an example of a board, and you can see that it's drawn blue boxes around all of the moth species, and then it's attempted to put a classification. It puts down the Latin name, and then you can see a little percentage score as well, and that's how accurate the classifier is, that it's identified it correctly to the correct species. And just to show you a couple of examples, we've had this deployed this year over a program called Ag Zero, which is looking at sustainability of farming methods in the UK, and there's a biodiversity element to this program. So we've been deploying these systems over a lot of different farms across the country, where they're either implementing agri-environmental schemes or they're not doing a standard control farm. And just a very, very brief slide to show you some of the things that we were able to see. So with this sort of wide temporal monitoring that I was talking about that's important, we can actually see here, you can see the red is our control farms and the blue is our agri-environmental farms. So you can see a lot more moth diversity. This is actually an abundance slide in the agri-farm versus the control, which is nice to see. And then what's very interesting is that we can also see that if we look at temperature, we can see that moth diversity it tracks temperature almost perfectly, and you can see the amount of diversity we have of species of moths in that September heat wave at the end there. And then we also have this system out on a project called Amber, which is a project that I manage. And this project is deploying these systems across, we've got 40 different systems being deployed over 6 tropical hotspots worldwide, just with the aim of trying to build those classifiers for regions where there's lots and lots of biodiversity and not a lot of work going on. And so future developments for this system, because we do recognize that it's a very new system and there's a lot of issues with this system so far. So just to briefly run through some of the future developments, we're installing telemetry on there, which mean the data, rather than us having to go back and collect the data can be sent back over the network. And with telemetry also comes the ability to run remote health checks. So every day the system will be able to tell us and send us a little file back telling us if it's working, if it's turned on, if it's taking images. And the benefit of that is if you leave, imagine if you leave a system out for 1 year and you come back a year later and you found out it broke on day 2. So we need to know if the systems are working. And this goes for all tech, not just for the AMI system. We're adding on the audible and ultrasonic acoustics. We're looking at alternative power sources because obviously it's difficult to power these systems under, say, a closed canopy with solar. And then we're also developing an AMI Mini, which will be a mini version that you can essentially put in your backpack and hike up a mountain with, because at the moment these systems are very large and you need really good accessibility to deploy them. And on the software side as well, we're building all data pipelines into the cloud, so we don't have to download and analyze all this data on computers or hard drives. As the data is uploaded to the cloud, the software and the analysis will essentially run over the data. We're installing edge processing, where these the models, the classification models, will actually be on the device. And we're currently doing lots of comparisons of the manual versus automated methods to see if we're pulling in different species and how these lights are also affecting wildlife. And we're constantly working on the improvement of the classifiers with more data that we get in. And so I just want to move to something a little bit more fun at the moment. So I've been doing quite a bit of work over the last few years on robotics, specifically on robotics and bioacoustics, but this can be applied to any different types of sensors. And I wanted to show you this because I think often peatland environments can be quite inaccessible. And one thing that robotics can do is help us access these really hard to access and difficult ecosystems without damaging the ecosystems we're trying to access. And I just want to show you a couple of examples that I've been working on over the last couple of years. So this here is called SailMav. So SailMav is it's an aerial aquatic hybrid robot, and what he can do is he can fly through the air, and you can see the sails up at the moment. Those are usually down when he's flying, and then when he lands on the lake, the wings turn into sails, and then he can sail around. And the reason we were looking at this robot is we were installing acoustic sensors on the top of the robot, and on the bottom of the robot a hydrophone that dropped down into the water when it landed. And we were able to record in a lake in Croatia across areas of this lake that had never been had recordings taken on them before, because they were too sensitive for people to actually go in, and the environment would have been damaged by going in and making those recordings. And so we were able to record in really sensitive areas such as reed beds and collect really important data on species there. And so with this system, this is just some information from soundscape analysis. We had one acoustic recorder on a buoy, which was in the center of the lake, and then we had our system, which was moving around. And you can see here we collected completely different information on species from the single location versus the system that was moving around the lake. And then even more high tech, this system here, I'll just explain it briefly. Often when we're taking recordings in locations, or even installing any systems, cameras, acoustics, we install them where we can access on the ground, usually a meter or so above the ground. And when we've got forests or ecosystems where the canopy is a lot taller, we're often missing that important information from within the canopy. So we designed a robot that could fly up into the top of the canopy, and it can essentially shoot little acoustic recorders into the top of the tree. So I'll just play this video, and what you're going to see a few seconds in is you're going to see this drone shoot a little arrow into the top of the tree. And what it's doing there is it's shooting an arrow with the acoustic sensor in the top of it into the canopy. And from this, we were able to record the shrub layer and the canopy layer. And we found a 30% difference in species from the shrub layer where we usually record to the canopy layer, and a very different soundscape. So just to show you how robotics can actually be used to increase our accessibility and help us to study in areas that we wouldn't usually be able to. And then just very briefly at the end, I've told you about how amazing technology is. So I now want to just go over some of the benefits and some of the limitations of technology as a whole. So I think technology, as I said at the start, it has that ability to give us that really wide temporal scale. We can record over days, months, seasons, and years, and really capture information that we wouldn't usually be able to from traditional monitoring. And we can also cover an increased spatial scale and coverage as well. It can provide us with increased accessibility to areas that we wouldn't previously be able to access, and it can provide us with that standardized data collection. I think one problem that we find a lot is that we're all using different methods to study in different places, and are those methods comparable over different spatial scales? Potentially not. We believe it can reduce bias in the data that we're collecting, and reduce the invasiveness of that data collection, and also provide us with the high resolution, verifiable and replicable data. In the long run, technology is cost effective because we're reducing those person power requirements, but obviously it does require an initial outlay. We think that technology can capture rare species because we're leaving it out in areas that we perhaps wouldn't have been previously able to study, and for longer periods of time. and obviously reduces that observer bias when different people will see different things when they look at the same picture. And one thing I really like about technology is that you can answer multiple questions and study multiple species with one data set, where sometimes traditional approaches are designed to look at perhaps one question with one specific species or taxa in mind. And it's also providing us with new ways to monitor human activity, such as illegal logging, hunting, and fishing. And now on to some of the challenges and limitations that I see with technology. So the initial outlay of the device costs is obviously quite hefty. The AMI system alone, one AMI system costs £5,000. You do manage to recoup those costs back if you think about the sort of time spent in the field overall, but it is a big initial cost. And also, these devices need to be robust. They need to function well. You don't want them breaking a couple of days or a couple of months in. And like I said, you need to know if they're broken. So that device needs to be able to communicate to you that it's still working. And there are issues with power supplies. For example, with the AMI system, we're developing the AMI Mini so that we can work under the canopy on battery power, but power supply is difficult and it is limited to these devices. And also, there's a lot of people are a little bit scared of technology. I think there's definitely a fear around tech. And there's also that expertise and interdisciplinary working, which we've got a long way to go with yet. And then in terms of the analysis, I think the interdisciplinary working is even more key at the analysis stage. But the metrics, so abundance and density is very hard to get with technology, because we don't know if that one species say if we get 100 calls in one night of a species, we don't know if it's one animal being very noisy or 100 different animals making a sound once. So it does make some of the metrics that we would want to use quite difficult. And I think, as Brian was saying earlier as well, technology does struggle to identify some very similar species to species level, and you might only get to genus level or even family level with some species. For example, the micro moths are very hard to identify, so we do still need those traditional collection methods and identification. Data storage is always a challenge. We're producing hundreds and thousands of terabytes of data, and there's got to be somewhere to store that. And there is obviously there's a cost to that, and there is obviously an issue with sustainability around storing that much data. And the big data, the analysis processing pipelines for big data is complex and requires a lot of expertise. I would question whether the functionality of the current metrics and methods we're using for analysis are suitable for all these new techie data analysis methods that we're using. We're using the same methods to analyze data. They're 20 years old, some of the statistical methods that we're using, and were designed for data that was very different to the data that's being collected now. So I think in terms of statistical methods and analysis, we do have some way to go yet to be able to analyze this data. And then in terms of species classifiers and AI, we have a long way to go to make sure that these classifiers are accurate. The annotation and collecting those label training data sets is very difficult and requires lots of time and expertise. The training of classifiers also requires a lot of expertise, and the validation of these classifiers. Is one classifier produced for one species in the north of England going to be suitable for the south of England, for example? And how well are these classifiers actually working? How accurate are they, and can you rely on the results that come through them? So despite technology being amazing in my eyes, there are some challenges and limitations, and these are just a few of them that I've gone through today. So if anyone has any more questions, yeah, please put them in the chat, and then we can talk about them more in the Q&A.
Fabulous. Thank you, Jenna. That was a brilliant introduction to some of these really interesting developing technological approaches for biodiversity assessment, and definitely a lot of food for thought there as well. So next up, the third in our lineup of 5 today is Nathan Thomas. Nathan is the Project Development and Community Engagement Officer for Lancashire Peat Partnership, and he's going to speak to us about their approaches to biodiversity assessment. So I will hand over to you, Nathan, to share your screen.
Hi, I'm Nathan Thomas. I work for the Lancashire Peat Partnership through the Lancashire Wildlife Trust, and today I'm going to be talking about the biodiversity monitoring we do across the Lancashire peatlands and our experiences of doing that. So firstly, I just wanted to briefly say why it's important to measure biodiversity from a trust perspective. So I've got a few short bullet points here. Now, historically, biodiversity hasn't always been a focus for peatland restoration. It's been more focused on things like monitoring the hydrology and the carbon sequestration potential. But I think it's important to measure biodiversity because it can be used as a proxy for both of those things. And also it has its own benefits in terms of things like natural capital. And in fact, we are now seeing a change with things like the Peatland Code, which are taking things in, like taking biodiversity into account now in the future, which is a good sign. It can also be used as a metric for getting things like peatland condition, so your baseline condition, say if you're doing something like a peatland grant scheme project, and then it can also be used to measure the project as its trajectory changes over time to see is your biodiversity increasing with the changes to the peatland that you've made or not, and then can inform future management because of that. Particularly from a trust perspective, it's useful as an engagement tool for people, for like the public. So a lot of the time the public want to know what plants and animals you're going to find on the site when they go to visit you. And the same with things like stakeholders, so landowners, they want to know what is on their land and then what will change through the peatland restoration project that you do, because that can get them engaged and then will hopefully convince them to then want to do more work and improve the biodiversity on their land in the future, which is a benefit to everyone, really. And then, so particular species provide functions to habitats, which again gives a need to measure the biodiversity for that site. So for an example, well, an obvious example is sphagnum. So the more diverse sphagnums you have on your site, forming those hollow structures, gives you an indication that the ecological function of sequestering carbon, growing peat, is happening on that site. So that's, again, another use for it. And then quickly, invasive non-native species, if you do a biodiversity survey, a veg survey, and you notice there is a conifer growing on your site from a neighbouring plantation, that will inform you of the management you have to do on that site in the future. Next, I'm going to move on to why do we use different biodiversity measuring methods? Again, just got a few quick bullet points that I'm going to try and quickly go through. Firstly, taxa and habitat specific. Particular methods you can use for one, taxa won't work for the others. So a really obvious example is you wouldn't use a bat detector to look at the biodiversity of plants in your area. Secondly, it can reduce, well, more of the technological biodiversity monitoring methods, things like what Jenna mentioned, can reduce the danger to your staff when compared to going and doing traditional biodiversity monitoring techniques on a site, especially when you've already re-wet it or it is in quite a remote area. And then this sort of feeds into the next one, really. It increases the effectiveness of the monitoring that you can do at a larger scale. So if you're using something like looking at aerial imagery or using bioacoustics across your site, you can do it on a larger scale more effectively than trying to do traditional monitoring techniques where you have to take out a team and go across the whole site and do them all individually. And then that also allows for passive data collection, which I'll talk more about on the next slide. It's also person dependent on what type of biodiversity monitoring technique you will use. So as Brian mentioned, a lot of the traditional biodiversity monitoring techniques are being lost. We don't have as much skills with identification across conservation anymore. But at that same time, there's a lot of newcomers that are coming into the area now that have things like GIS skills and drone flight skills and can do things like read multi-spectrum data from satellite imagery, which is all very useful. So it's about being able to adapt to what staff you have available at the time to use for that particular project. And then things like work and project budgets and staff time will inform what biodiversity monitoring technique we will use. So for example, if you had a huge area with a lot of funding from a project, you could use very expensive monitoring techniques. Whereas if you have a sort of legacy site that you've owned for a long time where there's no initial projects going on right now, but you still want to monitor the biodiversity, you might use a less technological cheaper alternatives such as traditional identification for biodiversity, which is still valuable. Then, again, sort of already been talked about this, we have a constantly changing and wavering volunteer base. So, for example, on one of our lowland sites, we have just lost a Friends of group who historically knew a lot about the site and did a lot of our vegetation surveys, but now that all they have gone, we have lost that incredibly valuable resource. So we have to overcome that somehow, and we can't always just find a lot of new volunteers in such a small space of time. So we'll have to probably adapt and use another technique that will be as effective, but with less resources, basically. And we've also got a birder who's been on one of our sites for about 60 years, and he's just been voluntarily giving any of the birding data that he's collected, which has been massively useful to us. But as he will disappear eventually, I don't know what we're going to do to replace him. So we need to find some sort of effective biodiversity monitoring technique to replace that. And then lastly, I put this grant specific, basically to say that when applying for grants, they will find it more appealing if you have a multitude of biodiversity monitoring techniques and some more interesting ones or innovative ones as well. Now, next, for these next two slides, I'm going to talk about two different sites that we have that we cover for peatland restoration. The first one is Darwin Moor, which is an upland site. It is funded through the Nature for Climate Peatland Grant Scheme, which is a project where we get 2 years of funding to do restoration works, and through that we want to monitor it for various different taxa. It's an open access moor, was a previous grouse shooting site. They still have the rights to shoot on there, but although they don't shoot on there at all now. And the biodiversity monitoring that we do use on this site includes your traditional vegetation surveys. So this picture here shows a 2x2 meter quadrat that we used for the baseline survey for the Nature for Climate Peatland Grant Scheme. Traditional veg surveys are useful because they are quite robust. You get a lot of data, but you do need to have someone who has those vegetation identification skills. You need to actually go on site to do that. It can take a lot longer than some other techniques. And also you need to go up with at least 2 people, because one will act as the scribe while the other person identifies all the plants that are up there. It can be things like weather dependent, and if it's a difficult site to get to, there's all those risks as well. But again, it's still very useful, and it's sort of your bread and butter in conservation. Then we have bioacoustic monitoring, which Jenna briefly talked about earlier, but this is specifically audio moths. And the reason we've done this on Darwin Moor is, as I mentioned earlier, it's an open access moor. It has about something like 500,000 visitors per year. And it's been noted that from the traditional sort of BTO walkover surveys, where you just walk and you just monitor what birds fly up as you walk past, they think that you don't get a true reflection of what that bird biodiversity is up there, because the birds are effectively tamed because of the amount of visitors that come up there every year. So we feel that using bioacoustics, a passive form of monitoring birds, will be more effective on this site, because we'll leave it up for 3 weeks on 3 separate occasions throughout the breeding bird season, and they'll record for 2 hours at a time in dawn and dusk. And that won't be dependent on you frightening the bird to fly up so you can record it. It will just pick up all of their calls, and I'll only have to go up there once to set it up. I'll leave them up there for that 3 weeks. After 3 weeks, I'll go and collect the SD cards, which are in each of these small devices. Then I can take it back to my laptop, put them into a software like this Raven Pro, which will create sonographs similar to what Jenna just showed, and hopefully using things like AI, we can quickly analyze all that data, because that will be quite a wealth of data to go through. And it will be able to pick up how many individual calls of each species were recorded over that time, which will be a lot more efficient than doing those traditional BTO walking surveys. But we're yet to see how that will work. And then I want to briefly talk about the remote sensing data that we're going to try and use. Now, I assume this GIF is moving. It's showing monthly aerial images from the Sentinel-2 satellite, and it's showing the normalized difference vegetation index. Now, that basically picks up plant health, and also can be used to measure biodiversity, especially when you know what habitat and what the hydrology is like there. So these sort of darker green areas usually indicate where the plants are more healthy, and you can see the differentiation in colour is showing different plant communities. Now I'm going to couple that with the vegetation data that I collect through the traditional veg surveys to basically cross-validate the two different techniques to make sure that they're picking up sort of the same data. And then hopefully in the future, I will then be able to use this remote sensing technique more efficiently in the future, because you can use it in such a large scale way, and also it's open access and it's free and it's constantly being collected by the Sentinel-2 satellite. So whereas I have to collect the data for the vegetation surveys now, this is being collected anyway, so I can analyze it years in the future or something like that. Now I'm going to move on to the lowland sites. Sorry. Now these are the Greater Manchester mosslands. These are about 5 or 6 sites, all owned by the Lancashire Wildlife Trust. They've either been previous peat cutting sites or agricultural land. And the biodiversity monitoring that we've used across these include, again, your traditional vegetation surveys. We'll talk about them again. But also we've used biodiversity monitoring to reintroduce the large heath butterfly, which is one of the main things the Wildlife Trust is known for. So to do that, again, we did traditional vegetation surveys for the initial peatland restoration works that we did. That was then used as a feasibility study to see if the food plant and the habitat planned for the large heath caterpillar and butterfly were there. So we were looking for Eric tetralix and hare's tail cottongrass. Then we also did butterfly surveys, as you can see in the bottom right corner there, to see if there would be any competition with the large heath and if they'd actually come back through peatland restoration themselves naturally, which they hadn't, unfortunately. So then in 2020, they were reintroduced to Astley Moss through working with Chester Zoo. And then following that, we have kept up with the biodiversity monitoring for the butterflies themselves, again, looking at are the butterfly populations staying stable, are they growing, are they shrinking, and is their food plant still available, is the habitat itself still suitable for this species? And then another form of monitoring we're doing, again, is bioacoustics, but instead of looking at birds in particular, we are looking at things like the bog bush cricket, which was a similar story to the large heath, was once very prominent in this area, but through things like industrial revolution and intensive agriculture, it disappeared from this area about 100 years ago. And a lot of the original surveys we did for this species were just visually trying to look for them and trying to listen out for them, which is really quite difficult, because when you do find them, they look very similar to another species called the Roselle bush cricket. But when you use a bat recorder, you can actually hear them very easily, so you don't always have to find them to know they're there. And also the difference in the call between the two species is very distinct. And what we were able to find out was that there was a lot more Roselle's bush crickets than we thought, and the species that we thought were bog bush crickets in fact weren't. And it gave us insight into the competition that they were having, and also informed the restoration and the management of some of the sites that we needed to change in order to get the bog bush cricket back. Another thing we use in some of the lowland sites is LiDAR, and we get LiDAR imagery using drones. So these images will get an understanding of the microtopography and the vegetation structure which is across the site, which is a good use. It's a good proxy for biodiversity, really, because you couple that with imagery, and then you also do like to cross-validate that with actual vegetation surveys. And instead of just doing one flyover with the drone, we'll do them seasonally to see how that microtopography changes, because again, what you might think is a sphagnum hummock after one flyover might disappear, say, in the winter, which a sphagnum hummock wouldn't usually do. They would change in a small degree, but nothing large. So again, that's another good way of using tech to get a proxy for biodiversity. But again, we do like to couple that with traditional vegetation surveys. And then we use environmental DNA. So I assume everyone knows what eDNA is, but to quickly go over it, it's a way of taking samples from bodies of water to see what DNA is in there, and that usually gives you an indication of what species are present at that site. So particularly what we've used them for in the Greater Manchester mosslands are for the reintroduction of the white-faced darter. Now, the white-faced darter, again, is a similar sort of story to the previous two insect species I've mentioned, was once prevalent in the whole of the UK. since things like climate change, intensive agriculture. They are now in two very disjunct populations, one in sort of the Midlands of England and then up in northwest Scotland. There are smaller populations in Cheshire and Cumbria, but we would also like to create another stepping stone site for them in Greater Manchester, with the idea that once there is suitable habitat there for them, the population in this sort of, well, the Midlands of England will be able to use it as a stepping stone site as they move more northward due to climate change impacts. So particularly what we did was we took eDNA samples of the bog pools across the sites to look for invertebrate biodiversity in terms of looking for prey species, suitable prey species for the white-faced dart and nymphs as they'll grow up in those bog pools. The results were mixed. We found that on some of the sites it was excellent and would be ideal to reintroduce the white-faced darter, but for a lot of the other places, as we're still relatively early in restoration on those sites, they're still not quite adequate enough for reintroduction of the white-faced darter, sorry. Then we also have been using eDNA for our paludiculture trials. Now, this is a picture of Typha, which is a crop we're trying to grow on some of the old traditional agriculture sites that we have. We have since bunded up those places, raised the water level and created wetter farming sites in order to sequester carbon and keep the water there for longer. Also, to give a financial income for these landowners, we've trialed growing Typha. And what we also want to see at the same time is, are these wetter farming practices increasing the biodiversity of those areas in terms of microbial communities and invertebrate communities that are in those pools that we've created? So again, similar to what we've done for the white-faced darter, really, we just want to see what invertebrates are in there. And then just to move on to say that within the Lancashire Wildlife Trust, we are still at the early stages of using these extra, more technological biodiversity monitoring techniques. And also these newer technologies for biodiversity give us scope for achieving data collection on a much larger scale than what we currently can do just using in-person biodiversity monitoring techniques, especially in the face of wavering volunteer bases and loss of ID knowledge. But again, it is also important that we blend traditional techniques with new techniques to cross-validate them and increase our overall effectiveness of biodiversity monitoring, because we need to know which are the most effective in different sites and how we can go forward in the best way possible. And also, we want to make sure that our monitoring that we're currently doing now has wider benefits in the future. So we are collecting a wealth of data that can then be used through partnership in the future to anyone else who might have questions or objectives that they want to meet. So we want that backlog of data that can be shared and used for overall conservation and a wider benefit than just ourselves. And that's it. Thank you very much.
That's brilliant. Thank you, Nathan. And it's really nice to see how you guys have been using that blended approach with some of these developing techniques, but still, valuing the importance of traditional approaches as well. It's fabulous. So again, if you've got any questions for Nathan, pop them into the Q&A. And I will hand over now to Jessica Gauld. So Jessica is a PhD student at the University of Manchester. And she's going to take us into the world of paleoecology. So if you liked our species showcase on Test Date Amoebae, then this is the talk for you. And thanks to Jessica for helping the Peatland Programme to produce that one. So over to you.
Hello, my name is Jessica. I am a third-year PhD at the University of Manchester, and I will be talking to you about paleoecology, how it can offer an alternative perspective of peatland biodiversity, and the importance of collaborative working to truly realise the potential of this research in supporting peatland management. So, what is paleoecology? Well, as a short overview, paleoecology is the ecology of the past. It is a study of past ecosystems and the interactions of organisms within those ecosystems and with the wider environment. It covers long timescales that can potentially span a few decades to hundreds of thousands of years, or potentially even longer. And through paleoecology, we can reconstruct these past ecosystems, see how they changed and developed over time, and make inferences about why those changes happened, such as due to climate change or human-driven pressures. And as we can't literally travel back in time to make those observations, paleoecologists use fossilized biological and geological material found in sediments such as peat as proxies to reconstruct these past environments. So this diagram will be just a snapshot of some of the fossil material used as proxy records in paleoecology. Peat is an excellent archive of this fossil material, as its accumulation lets us see changes over time, and its waterlogged conditions make it great at preserving organic material. This diagram shows how we might take a peat core through the centre of what is, in this case, a raised bog. An example section of this core is shown here on the left. And a core such as this may be sampled at various intervals to tell a story of the ecosystem over time. There are many different proxies we can use that will tell us different things about our peatlands, and we can use combinations of these in multi-proxy studies to give a more comprehensive overview of the site and the drivers of change. So as an example, we can use fossilized pollen and plant macrofossils, which reflect past vegetation. So these may act as proxy records for regional and local vegetation change. If you were interested just in the local vegetation of this particular raised bog, we might prefer to use the macrofossils, as the pollen grains are much, much smaller, and as shown on the diagram, they can potentially travel much further distances. So they could tell a story that covers a greater spatial scale than the larger and the more mobile macrofossils. We can also use proxies within our record to infer environmental conditions and give context to the changes that we see in the vegetation or the other species and communities. So as an example, we can use these tiny life forms known as testate amoebae, which have already been introduced by Brian and which live in watery environments such as peatlands. We know these are quite sensitive to environmental conditions, particularly hydrology. These organisms are part of our peatland microbiota, and we can use them to infer how wet or dry the bog has been through time by using the morphology of their shells to identify different species at different levels of the peat, and then model the water table levels using a transfer function approach. And this can be a really useful thing to use alongside our plant macrofossils, because as we know from our modern peatlands, changes in the water table and the hydrology can impact the vegetation. This is just a very quick overview of many possible analyses we can perform in our peat cores. The emerging use of sedimentary ancient DNA, which is similar to eDNA as described by Nathan, is a tool within paleoecology which could give us a broader understanding of past biodiversity, including animals and more of the microbiota, as well as the vegetation that we've seen here. And other proxies such as charcoal and elemental composition could tell us about fire history or the increase of heavy metal pollution and allude to the rise and changes in human impact on peatland ecosystems over time. The availability of dating techniques such as radiocarbon dating allows us to compare with other sedimentary records and place changes that we see in the ecosystem within the context of known climatic events. So on its own, this is all very interesting stuff, but how does this relate to present-day peatland biodiversity and the management of these environments? The primary advantage is that paleoecological study can extend or supplement historic monitoring records and gives us a much longer temporal view of our unique site history. This difference in temporal scale is exemplified by the timeline on the slide, which shows the potential reach of a paleoecological record compared to a historic record, as a historic record may only go back 100 years or so, and it may be incomplete. It also shows some of the main climatic events that happened during the Holocene, which was a major period of peatland formation and which occurred over the last 11,700 years. For understanding and managing peatland biodiversity, this gives us a way of gaining context for the modern peatland site and the state of the modern site, and what the most appropriate long-term targets and expectations for its recovery might be. Paleoecology can add a longer-term perspective on what might be considered a natural state for the peatland and provide context on the impacts of past changes and pressures and how the site and the biodiversity might then respond to future environmental or human-induced pressures. In terms of what is natural, paleoecology allows us to see what our peatland biodiversity was like before intense human intervention altered and damaged them. This may help us avoid shifting baseline syndrome when setting our reference states and biodiversity targets for restoration. Shifting baseline syndrome describes situations where the baseline for what is considered normal or natural in an environment gradually changes over time and generations due to a lack of information or memory around the original or the true pre-disturbance state. A consequence of this is that management targets might reflect a false perception of what the natural conditions of the site were, and instead still reflect the state that had already been altered. There are, of course, other factors to consider when setting target conditions, but the longer-term perspective on pre-disturbance ecosystems might be a valuable addition to an evidence-based management approach. Paleoecology can also grant us perspective on ecological processes that occur over much longer timescales than typical monitoring efforts might cover. You can see that the climate of the Holocene was far from static, and through their development, peatlands may have already weathered a number of climatic fluctuations. And against this background of climatic instability was, of course, the growing impact of humans on the environment through the rise of agriculture and industry. So it makes sense that our peatlands were also not static systems, but have shifted and evolved through time to either resist, recover from, or change under various pressures. The current state of the site might reflect the legacy of human and environmental pressures that, again, might span longer timeframes than what is captured by monitoring or historic records. Such sites might have already been existing in a less desirable state even before the more intense climate change and human interventions of recent centuries. And this could have implications for a realistic approach and timescale for their recovery today. Adding a paleoecological viewpoint could be a really important part of seeing the full picture. So there is a lot of identified potential for paleoecology to be an additional information source for environmental management and how we view biodiversity on peatlands. However, paleoecology in its traditional form is not always easily translatable into practical action. The figure on the right here illustrates a section of a typical pollen diagram, which is a way of visualizing changes in the pollen data over the depth of a core. And I think it's fair to say that if the goal is to turn the research into practical action, figures such as these and traditional academic papers are not always the most intuitive way of doing so. Furthermore, research priorities do not always align with management priorities. Paleoecological studies may cover a much broader spatial or temporal scale than would be relevant in the management of a specific site, and research questions won't necessarily lead to feasible and management-driven conclusions if they aren't designed that way. For this reason, collaborative working is key to making science actionable. This example workflow illustrates how the different steps in the research process may be framed in a more collaborative way, with academics, stakeholders, practitioners, contractors, each contributing at each stage to reach actionable outcomes. Paleoecologists are likely not experts in practical conservation and site management. Therefore, allowing the process to be a partnership rather than a one-way transfer of knowledge from research to practice means that the research may be framed within the decision-making context. And this means that the results are more likely to reflect the needs of the site and the feasibility of practitioners to implement recommendations. You can see that this process starts before the research goals are even set, with creating good working relationships. This is done through, of course, communication, but more importantly, transparency about the capacity, the capabilities, and the resources of all those involved. This means identifying early on what the paleoecology can contribute based on the current priorities for the site and what the constraining factors might be, such as targets, funding windows, and project timelines, and what paleoecology can realistically deliver within those framing factors. This is also the time to set expectations for the project in terms of the nature and the frequency of communication, the responsibility of all those involved, and how and when the results will be produced and used. This stage does link quite nicely to stage 2, as jointly identifying the value of paleoecology will lend itself to conversations around creating specific goals that align with the goals of the overarching restoration project. Although collaborative working throughout the process is important, the first 2 stages really are particularly significant for setting up for success. Waiting until the results have already been produced to start that collaboration might be a little bit too late. Collaboration remains key throughout the remainder of research, including the fieldwork, lab work and data analysis, and the framing and integration of research as practical management steps. Maintaining communication and regular updates, as well as shared responsibility where possible throughout these stages, can be important for maintaining a sense of ownership and buy-in from all involved actors. This is particularly important when implementing that research into practical action. So although this is in part the responsibility of the paleoecologist in how they present that data, creating the time and the space for workshops to jointly discuss the management implications can be key for producing feasible outcomes that integrate both perspectives. So with that, that is the end of my presentation. Hopefully that has given you a snapshot of some of the ways that paleoecology can lend itself to peatland management and why it's important for academics and practitioners to really work together from the beginning of the research process to realise that potential. If you have any questions, please feel free to get in touch, and thank you.
So this is the Peatland Programme's very own Joe Anderson. So he joined us at the end of last year as our biodiversity crediting project officer. And Joe is going to talk to us about some exciting developments for the Peatland Code, which is the Peatland Programme's voluntary carbon finance standard. So
great. Thanks, Jane, for the introduction. So I'll be presenting on the biodiversity crediting project under the Peatland Code. As Jane said, the Peatland Code is a standards and framework for quantifying the carbon emissions that are abated through peatland restoration within the UK. The aim of this project is to create new nature finance pathways for peatland restoration. And for those of you who are in the UK for this presentation, this is a Ferns-funded project, so it is funded through a collaboration between NatureScot and the Lottery Heritage Fund. So a little bit of background, there's a ton of benefits to peatland rewetting. We've heard about quite a few of them already today. The image to the right shows all the different types of ecosystem services that are affected by peatland rewetting. However, when it comes to nature finance, which is ways for private equity to support ecosystem uplift, the current market focuses mainly on carbon, and that's completely understandable. There's a pre-existing market for carbon. It's really easy to quantify, and there's a ton of carbon emissions that can be abated by restoring healthy peatlands. However, there's just so many more benefits than carbon to look at for peatland rewetting, and sometimes the carbon market isn't actually enough to fully fund the process of peatland rewetting, even with public funds included. So now there's an emerging biodiversity crediting market, which, like the carbon market, can sell sort of units of ecosystem service, in this case being biodiversity uplift. It's a really new market. Even some of the leaders in the market on a global scale are just beginning to start trialing methods this year. And currently the market is really aimed at global frameworks. And that's one of the places where the UK Peatland Code has an advantage, because we are only working with UK peatlands. So our biodiversity crediting framework can be very specific to these habitats. This program also is in collaboration with the Woodland Carbon Code to develop biodiversity for UK woodlands, but we'll just stick to peatlands during this presentation. So the project goals include market research and experimental design. So we want to make sure that we're designing our framework to be in alignment with existing market trends, just so that as the market evolves, we are still meeting the rigor that other larger crediting organizations are creating. We are working with teams of peatland experts as well to identify key indicators. So we want to make sure that what we're measuring is appropriate to UK peatlands and appropriately tells that story of biodiversity uplift through habitat restoration. This spring and summer, we are piloting a trial MRV. So we have some sites where we'll be implementing monitoring. The aim is to collect data, see what type of outputs we get from that data, identify any challenges, so project developers and landowners who are trying to do peatland rewetting can inform us on what maybe just doesn't work in the field this summer. And from there, we aim to develop a crediting framework within the Peatland Code by next year. So there are several challenges that we've run into. As you've heard across this presentation, biodiversity monitoring is complex and very nuanced depending on the conditions. One of the big issues we've run into is that a lot of traditional biodiversity metrics don't necessarily increase in value with habitat restoration in peatlands. So species richness and species diversity, taxonomic dissimilarity, these are really common ways to measure biodiversity uplift. However, they just don't translate to peatlands, given how unique the habitat structure of healthy peatlands are. For these metrics to be relevant in nature markets and really activate private finance, the data has to be asset grade and auditable. So this is that gold standard data that Jenna was talking about earlier. The indicator selection is, as I mentioned earlier, a really key part of this. So it has to be specific to the UK peatlands, but we want to see which indicators are the most sensitive and the most indicative of the process of habitat restoration. That way we know where to focus our measuring and monitoring processes in order to really make sure we're getting that picture. And finally, this has also come up several times during this presentation. The price of biodiversity monitoring can often be very high. And for this program to be successful, it cannot be cost prohibitive for early adopters into this really novel market. So making sure that we're approaching this in a cost-effective way. Ultimately, within a biodiversity crediting framework, we won't be doing complete ecosystem assessments, but we'll be identifying the cost-effective ways to monitor the indicators that really best tell that story of habitat uplift. So here's some of our approaches to these particular challenges that we've been facing. With the biodiversity metrics, we're hoping to develop a peatland scale for UK peatlands from completely degraded to totally pristine, using the indicators that we're developing. And so that way, when a new site baselines their current habitat conditions, we'll be able to sort of place that site along a scale. That will give us the advantage of being able to predict what types of habitat uplift in terms of percent improvement in metrics are feasible because we can see where it aligns across that entire scale. And then subsequent measurements after peatland restoration has occurred can be placed along that scale, and you can get a percent improvement on your peatland. For the auditability, we are focused on the creation of a transparent data pipeline, which does prioritize automated and digitized monitoring strategies. We've heard a lot about that today. However, we will always include ground truthing, especially because some of these technologies are really new. So we want to make sure that they're working and that the measurements that we're using are accurate to the site. Coming back to indicator selection, we've already worked with a team of UK peatland experts. And then after our piloting, we'll use that site data and multiple external data sources that we have for UK peatlands to define what is a positive and what is a negative indicator along different points on that scale. So you don't have to measure every single thing to be able to tell the overall biodiversity story. And that way you can look at it in a more nuanced way than just species richness or taxonomic dissimilarity. You'll be able to really define what shows positive improvement and what shows negative condition indicators. And finally, for the cost, we're looking at exploring packaged biodiversity monitoring services. So this is biodiversity monitoring as a service that can be purchased by project developers, landowners, etc. The benefit there is that these organizations specifically do biodiversity monitoring, so they can sometimes drive down the price either through economics of scale or just having all of the expertise needed in one location or through one service. And it reduces any technical skills required for entry. We have talked to some project developers who want to do this in-house, but we've also found services that the project developers and the landowners can focus on the peatland restoration and rewetting, and then for a fee, have all of the biodiversity monitoring completed. And like we saw earlier, some of that biodiversity monitoring data is super complex to process. And so that takes that onus of responsibility off of the project developers to process all that data. So this project only started in December, so we're about 4 months in. Right now, we've identified the indicators that we want to use, and we're in the final approval process internally by getting project partner approval. We also have a team of ecologists from the SRUC who are reviewing it to make sure that we're not missing anything, that we're making sure that appropriate indicators are captured through our metrics. We're in conversations to potentially align our MRV, which for those who are not familiar is measuring, reporting, verification. So the structure of how we collect this data and verify it. With the Operation Wallacea methodology, this is one of the first methodologies introduced for biodiversity monitoring and accreditation. They are UK based, but they aim to be a globally applicable system, which of course implies a certain degree of flexibility. So we've already talked with the chief writer of the Operation Wallacea methodology, and the trust that owns the method keeps it open source and is really open to collaboration with external organizations. So we're optimistic that we can work in an iterative fashion with the Operation Wallacea methodology to make it specific to UK woodlands and peatlands. That creates the advantage in that there will be a really clear framework for how biodiversity monitoring and accreditation can occur. An added bonus is that because we will have this specific methodology for collecting biodiversity data, there's the capacity for multiple sites to compare their biodiversity data. So you can get a better idea of which management methods offer what types of biodiversity uplift, or you can legitimize the biodiversity uplift that you're doing on your site by being able to compare it to multiple other sites using our biodiversity crediting protocol. We have some pilot sites identified. We're just working to finalize the contracts and budgets. So hopefully by May, we can start actually collecting our biodiversity data. And finally, we're working on a market research component where we're contacting carbon credit buyers and sellers who have historically worked with the Peatland Code to get an idea of what would attract them to be early adopters. The biodiversity crediting market, while it's expected to grow very quickly and be close to the levels of the carbon market within this decade, there's still a lot of uncertainty just because it's a brand new market, and the concept of reducing biodiversity to a single metric of percent uplift requires a lot of robust verification before it's actively accepted from a buyer perspective. But we're hoping to get as much of that information as we can now to ensure that what we develop works well in the long run. If you're interested in discussing this further outside of the Q&A section, there's my email address, and I'm happy to answer anyone's questions through my email or at the end.
That's fabulous. Thank you so much, Joe. It's really great to see, even in the short time I've been with the programme, all of the sort of progress that's being made with Peatland Code. And let's move on then to our Q&A. I'm aware that Jenna has to leave us a little bit early, so we'll start with questions for Jenna, I think, if that's all right. And we have a question from Tanya Milivita. Sorry, Tanya, if I've pronounced your surname incorrectly. She has a question for Jenna on, so the acoustics, the mapping of acoustics. Can you estimate the abundance of species as well? For instance, sort of X number of tigers per hectare, etc. And how do we approach silent animals?
So for abundance, you can't measure abundance, no, is the answer. Because like I said in the presentation, you don't know if that animal that's calling, all those 100 calls are one animal or 100 animals. What you can do is it reduces the coverage, but you can increase the amount of audios in an area, and you can essentially triangulate. So if you had 4 audio recorders and one animal was calling in the middle, or 5 animals were calling in the middle, and the distance, the time that it takes for the acoustic signal to reach each audio recorder essentially allows you to triangulate the individual, and then you can estimate the number of individuals. But that requires, it's very tech heavy, and it requires a lot more audios. So you study on a smaller scale, but you get more information, if you see what I mean. So in that way you can. The simpler way to do it is that we know for every species that we've done the calculations for, there is a direct positive correlation between number of calls and number of individuals. So you can see trends and patterns, and you can say that it's increasing or decreasing, but you probably won't be able to put an exact number on it. And for silent species, acoustics, no, but then that's when you turn to other methods such as cameras, eDNA. There's lots of different methods that we can use.
Brilliant, thank you. We have a question from Matthew Lane for you. So he says, sorry if I missed it, but how accurate is the AMI plus at identification? So this was in regard to the sort of confidence percentages slide that you had up.
So we're still working on that, and these classifiers are very new, so the system's very new. It's only a couple of years old, and it's still undergoing constant development. It's pretty good. I'm not going to put a percentage on it, but it is sort of, it's 70 plus at the moment, 70+%, possibly even higher for a lot of species. So it is quite good at identifying. That's in the UK and in Europe. Obviously, we're building classifiers overseas, and this is where it gets very difficult with 20,000 species of moth in one small country. And it's obviously better for the macro moths than it is for the micro moths. It can do macro moths very well. Micro moths are a little bit harder.
Yeah, and I guess it's with a lot of these things, the more data that comes in, the better the machine learning gets with this. So the more information
Exactly.
we have, the better these approaches become. Yeah. So there's a comment really from Jackie McBurney, who said, you know, she feels that these technological devices are a good supplement to human surveying. But the percentage of ID accuracy could be an issue, as we've discussed. But I guess it's a bit of a philosophical question in a way. Are we heading to the point where we won't need ecologists on the ground anymore?
I
would
that one
imagine
might
most
come
people
up.
disagree with that one, but it'd be interesting to hear your thoughts as the technology expert.
I think at the moment, we absolutely need the traditional methods. We're a long way off technology being able to replace anyone. And that's not really a stage that we want to get to either, because I think it will always be complementary. For example, with the AMI system, we attract lots of different moths with the type of lights that we're using, but there's, we need to do the comparisons. But what we're seeing so far is that we're attracting different moths. A lot of them are the same, but we're attracting some that aren't caught with using the manual methods, and vice versa. So I think it's more about adding to and complementing rather than replacing. But obviously, as these new methods come in, it might reduce the need for other methods. I can't say that sort of tech isn't going to replace some things, but I think we'll start to see the reduction of traditional and the increase in tech, but never the replacement.
Yeah, totally. There's another question for you from Kathy Atkinson. Is the AMI moth classifier open source? Is this something people can access?
So it will be. It's been sort of 2 years in development, this classifier, and unfortunately it's been buried under hundreds, maybe thousands of lines of Python code. But it's finally at the point where we have a web app that's been developed for it by one of our collaborators in the US. So the web app is undergoing trials at the moment by us, and then once all the little issues are ironed out, essentially it will be open source, and anyone could upload a moth picture to it and then could get a classification from that. It depends in what region of the world. If we're talking about the UK, it will be available and pretty good, but then if it's for other countries, the classifiers either are non-existent or very early stages of development.
Yeah, I have a question for you actually. How do you think this sort of compares to things like iNaturalist? Because I've used that quite a bit for moths. And I just wonder, yeah, if you've got any ideas on how they compare in terms of accuracy.
So I'm not sure, and it's not something that we've looked at yet. But all of our data comes from GBIF right now, so it should be quite similar.
Yeah.
I'd say the accuracy would probably be, well, I'm not sure. I'm not sure how iNaturalist does it. But because ours is a bit more specialized and has probably had a lot more work done on the algorithms, I would imagine that it would get to the point where it could exceed something like iNaturalist.
Yeah, we just need some technology now to dissect out genitalia, and we've got everything we need, haven't we? Fantastic. Thank you, Jenna. I'll move on now. We'll go back to Brian, actually, if Brian is still with us. I've got some questions for you, Brian. So there's a question from Lindsay McKinley. And they say, is the condition of bogs a reliable indicator of invertebrate population health for those who don't have or can't find anyone with invertebrate expertise?
I think the answer is yes, but. The but being current health doesn't tell you about past history, and some invertebrates may become extinct in the past and not been able to recolonize. Less of a problem possibly in the uplands where habitats tend to be fairly contiguous. But if you're looking at valley mires, raised bogs, and surviving intact fen habitats, then those will have lost quite a lot of invertebrates, which won't necessarily come back quickly when the site gets into better condition. And the other thing to say is that every feature of a bog will have its own specific group of invertebrates associated with it. So bog pools, I mean, I did study decades ago and to show there was only about 25% overlap between pitfall-trapped insects on the tops of sphagnum hummocks and at the bottom of hummocks and on sphagnum lawns. So the different microtopography has its own set of invertebrates. So as long as you're measuring all the features of a bog, then you've got a pretty good surrogate for the invertebrates. But I strongly say try to find an invertebrate person to have a look as well.
Yeah, definitely. And this kind of links to our sphagnum showcase. We talk about microtopography and that kind of fine scale zonation that you can get, the huge differences that you see in hummocks and hollows and across. It's a giant landscape, isn't it, if you're a very small invertebrate? So huge differences for
and
them.
there's a big question for me as to whether or not hummocks actually restore themselves on restored cutover bogs. And I've seen some cutover bogs that have got marvelous sphagnum lawns and they stay flat and boring for decades at least, centuries. And the only surviving hummocks are the ones that have actually been there for several centuries and have been moved around a bit. So you may need to give topography a helping hand.
Brilliant. Then we've got a question from Rosie Snowden at Yorkshire Wildlife Trust. She's on their peat team. So she says, because the vast majority of our funding is for restoration work, we aren't able to carry out as much monitoring as we would like. So could you give a few key species that are indicators of good condition in blanket bogs so that they can then focus their efforts on these and do some targeted fundraising?
Part of me thinks I should pass this one over to Joe and say this is exactly what the peat scale is going to be doing.
Well, Joe will be listening intently to your answer, I should think.
Yeah, I mean, I was good to say that I'd probably do it in sort of 3 stages. So there's quick and easy vascular plant stuff. So you know which vascular plants you want, but there aren't many. So the difference between an intact or a really well restored site and a damaged site is probably only 3 or 4 plant species. Bring in the sphagnum and you increase that by a factor of 5 or 6. You've got potentially, as we were just discussing, a good range of microstructures and maybe a dozen or 15 sphagnum species in there. I happen to like lichen, so I suggest throwing the cladonias for the drier bits of the site. If you're doing invertebrates, it's really a question of what can you identify, and it'd be really good to have a quick baseline before you go much further to make sure the right species are there. I mean, I was thinking on the Yorkshire pennines, the big metallic green ground beetle, Carabus nitens, is a pretty good indicator of bogs in good condition. But we know it needs about a square kilometre of habitat to survive long term. So if you're part of an extensive area of habitat, it'll work. If you're dealing with small areas that have been restored within a wider area that's still degraded, then a species of that sort of size probably doesn't work.
Yeah, I think there's a related question to this from Alex Curry, who says floral biodiversity is often used as an indicator for peat condition assessments. And I wonder to what extent mega and microfauna might be able to be used in a similar capacity.
I think it depends on the types of peatlands you're looking at. So in blanket bogs, then things like breeding wader populations among the megafauna are probably quite useful. In the lowlands, the problem is you've got very few truly bog species. So I mean, it was interesting, both I and one of the other speakers talked about nightjars. Nightjars actually like their bogs dried out. They're heathland species that cope with the drier bits of bogs, but won't breed on pure raised bog or blanket bog unless it's got sort of dry tussocks in the middle of it. The invertebrates, I think, do give you a really fine resolution distinctions, and they also pick up geographic variation that you don't get in any other way. So I've talked a lot about the South Yorkshire bogs. In a British context, they are the nearest we have to Baltic bogs. 5 or 6 of the endemic species are species otherwise only known from Poland, North Germany, and the Baltic states. So that gives you an idea that this low rainfall area in the east of Britain has raised bogs very different from those to the west of the Pennines or in the southwest of England. And I think the invertebrates are the only groups that give you that small scale geographic distinctiveness, as well as the chance to understand each of the small topographic features. One of the other nice things on if you're looking at indicators on nature reserves and sites managed with conservation in mind, is the invertebrates will also tell you which direction you're moving in. So you'll actually see an increase in exciting heathland species if your bog dries out a bit, and then a decline in those, but a rise in woodland species as the birch and the sallows take over.
Yeah, well, you'll be pleased to hear, Brian, that Joe is definitely looking at using the invertebrates as indicators, and I've been flying that flag as well. So that segues in nicely, actually, to some questions for Joe, if you're still with us. Yeah, there's Joe. Miles Wilson has asked what monitoring techniques are being used for the biodiversity metric pilot trials, and are these being developed in collaboration with peatland programs carrying out active restoration, who often know best what does and doesn't work in a practical sense on sites?
Okay, I'll split that up into, I'll start with the second part first. So we are working with some project developers. However, we want to make sure that we're not creating like an unfair advantage for certain project developers over others. So a lot of the feedback we've gotten has been from people like one step away from the project developers. So the people who work with the project developers in the validation process or in the standard setting. Although that being said, we have a lot of field ecologists helping us develop it. So it's to that second part, yes, to the extent that we can without creating unfair market advantages for certain developers over others. In terms of the specific monitors or monitoring protocols, they're a little bit in flux, and I get this question a lot, and I can't actually say the specific plan until we have sign off from all of the different project partners, which should happen in the next few weeks. So if this is something you're very serious about, shoot me an email, and I can let you know when we have it, formalized. But it is going to look a lot at vegetation structure. Similar to what Brian said, the combination of the microtopography as well as the ground covers is one of the bigger indicators. Richard Lindsay is a really renowned peatland ecologist, and he has a peatland condition matrix that predominantly looks at the combination of microtopography and vegetation coverage. And it looks like we'll be basing some of our quantification off of that matrix. To whoever was asking the last question as well, Richard Lindsay's peatland condition matrix is a really great way to monitor peatland condition by just doing vegetation monitoring. As Jane said, we're definitely looking at terrestrial invertebrates. We're looking probably at something along the lines of meta barcoding with different types of traps, just sort of identifying which trap works the best. And from there, it will be splitting up all of the results from those traps into what's a positive and negative indicator, which is going to be a little bit different regionally within the UK. But there's a lot of UK-based databases, fortunately, that will help us sort of suss that out. And then we'll be looking at a few different taxonomic groups as well. There will probably be a degree of bioacoustic monitoring, and possibly eDNA sampling of soil or water. We don't have the sort of official monitoring protocol yet to release, but I hope that kind of gives you an idea of the direction that we're taking.
Yeah, qualifying statement from Joe.
Yeah.
Very much in development. So
yes, covering my tail as much as possible. Yeah, with like updates as of this morning, but that's the overall structure we're looking at. And because we already have on-site validation as part of the carbon accreditation process, we will have on-site ground truthing and validation. It's just sort of in development to what extent the validation process will occur on site versus just a really thorough overview of the data collected.
Thank you, Joe. So a question from Johnny Ritson for you. So are these biodiversity credits going to link to the UK government's biodiversity net gain? So it sounds like, I thought they have their own official tool, so it sounds like you're doing something outside this.
Yes, so partially because this is mostly funded by the Scottish government, and BNG isn't, Scottish government's doing its own thing. one of the challenges that we were looking at with BNG is because we're coming from a carbon background, there's this challenge of additionality, which for those of you who are not familiar, additionality means that for a carbon credit to sort of be validated, it has to prove that this project wasn't going to happen anyways. And with BNG, it gets into this sort of legal additionality in that because BNG is required, it wouldn't necessarily be additional because this habitat uplift already has to happen with biodiversity net gain.
Yeah.
So it's taking a different angle on the market. Part of that is just because the peatland code has a reputation for being a really high integrity player in the carbon market. And just to meet our standards, the rules around BNG don't really allow that. It's not to say that in the future the code might evolve, especially because biodiversity net gain is, I'm sure, going to change a lot over the next 10 years. The other piece is we're looking at the voluntary biodiversity crediting market, which has a really different approach than BNG. And even proponents of BNG will acknowledge that there's not that much actual biodiversity monitoring within BNG. It's more habitat condition assessments, which are very important and they're easier to complete. Whereas within the voluntary biodiversity crediting market, although it's nascent and doesn't have like the guaranteed market that BNG does, what we're measuring in the biodiversity voluntary market is percent improvement across a collection of biodiversity metrics per hectare. So the data resolution is a lot higher in the voluntary biodiversity market than in BNG.
Brilliant. Yeah, thank you for explaining all of that. It's a pretty complex marketplace, isn't it?
It sure is.
So we have another question from Manon Q, and they're asking, will there be credits for maintaining / not losing biodiversity once it's at a certain level, or will the focus still be on increase? And also, how are wider landscape factors accounted for in the crediting?
Yeah, great questions. So with the maintenance or preservation of biodiversity, there is a certain degree of precedence for that to be within the biodiversity crediting field. For the scope of this first year of us rolling it out as part of the peatland code, we're not necessarily considering that. The main shortcoming in biodiversity in funding for peatland rewetting is that it's just so much cost up front to change the hydrological cycle to return the peatland to its wetted state, which once that happens, a lot of the biodiversity uplift happens anyways. That's when the carbon starts getting really stable again. So the degraded peatlands that aren't wetted is where you're losing carbon to the atmosphere. So there's the potential to include that later, but that's not within the scope right now. Again, could happen later. And then the second part of the question, which is the wider landscape factors. This is something we're still trying to figure out. One of the habitat conditions that we're going to probably include to at least some degree in the final sort of multi-metric, which is the combination of all of the different biodiversity data pieces being combined to give you that percent uplift, is habitat connectivity. And what happens if you do all this work and then your neighbor just destroys the neighboring peatland or cuts down the forest? People probably won't do that with peatlands. It's more of an issue on the woodland side. But that is something that we have to consider. Again, one of the challenges with this crediting market is a different challenge called double counting. So let's say that your project increases biodiversity on a property that's sort of outside of your jurisdiction. If we tried to count that in the biodiversity uplift that we assign to your site, and then your neighbor decides to do a biodiversity crediting uplift project, there's the potential for that biodiversity uplift to be counted twice. So just within the current, because the current biodiversity crediting market is being heavily influenced by the structure of the carbon market, it's really tricky to try and quantify some of the biodiversity benefits of your ecosystem restoration on a landscape scale unless the entire restoration project on a landscape scale is part of this biodiversity crediting project. Those are really tricky questions. I hope I was able to answer them as much as possible.
Well, it kind of leads into a question from Rachel Maskell as well, actually, around stacking and bundling. So she says, yeah, it's coming.
Yeah.
There is a lot of uncertainty on stacking and bundling of ecosystem services, and the Ferns description of this project mentions both. So if this represents a percentage uplift, would you count this as a bundling of ecosystem services? As in just trying to justify to buyers why they should do or get more for their carbon savings? Or is it stacking and an option for selling the biodiversity uplift to a different buyer? If that makes sense. That'll make more sense to you than me, I'm sure.
Yeah, I can break that down. This question comes up literally every time I talk to anyone ever. And for those of you who are not as familiar with the landscape of this ecosystem service market, bundling basically means you would sell a single credit for an entire site. And so it would be carbon plus. So you would be buying this, it's typically at its foundation, a carbon credit that also has these added biodiversity uplifts. Versus stacking is you would create two separate tokens or credits based on two separate ecosystems that occur from this habitat restoration. So just for any of you who maybe didn't understand that question. And the short answer to this is we don't know yet. This project is funded for another year, so we'll definitely be looking into that, and we're trying to answer this with every single organization we collaborate with. On a global scale, the answer is not even clear how multiple ecosystem service markets will address this issue. If you have the answer, shoot me an email. There's really strong pros and cons to both. I have my own opinions on I think what would work best, but there are people much smarter than me who don't agree. So unfortunately, I cannot answer that question because it seems like nobody knows. But by March of 2025, we will have a statement.
We'll definitely have an answer. So see
Yeah.
you then.
Yeah, circle back.
So a couple more questions for you, Joe, before we wrap up. One from Nicholas Corker that says, how does background climate change factor into a biodiversity investment portfolio? Because surely it brings uncertainty to the equation.
Yeah, so I mean, uncertainty in general is something that, again, is a facet of the carbon market that's informing the biodiversity crediting market. Yeah, we don't know how much climate change will happen, to what extent that will have impacts on ecosystem degradation. So one of the ways that uncertainty is managed in the carbon market is what's called a credit buffer. So the project will either have an estimated amount of carbon that's saved or a measured amount of carbon that's saved, depending on if they do modeling beforehand. And not all of the credits are sold. There's a certain degree, typically in the carbon market, it's about 20% of the carbon credits accumulated by different projects are put into sort of a bank. And that bank can then be used by the standards body or the registry to sort of redistribute credits that have already been purchased, where then that biodiversity has been lost. So there's likely going to have to be a degree of credit buffers for the biodiversity market. There's some consideration. So we're not going to regulate how people do their biodiversity uplift practices on their site. We're just going to be monitoring a percent improvement and assigning credits based on that. So I can't remember who I was talking to, but another external collaborator was talking about future-proofing these landscapes for climate change, and whether that means the way that you implement your uplift looks a little bit differently, so that you can kind of create a landscape that has the potential to easily transition as you get warmer, drier summers, etc. Again, this is a little bit more of an issue in the woodlands and rainforests in the UK, of interventions you can do that create a habitat structure that is more amenable to climate change. Again, with peatlands, it's just a lot of rewetting. Sometimes you can plug sphagnums. but making sure that those moisture levels are maintained is kind of the best thing you can do for peat. But yeah, it brings a ton of uncertainty, and there's probably a way that we have to build in market frameworks so that uncertainty or loss of biodiversity as projects develop don't lead to a loss of market certainty on the buyer side.
And get restoring our peatlands quickly, I think, is another.
Yeah,
really,
the sooner that it's a lot easier.
They can help us with this problem.
Yeah, right. It's a lot easier to maintain something than to rebuild it. So if we can get these market frameworks up in time to get the peatlands wet before it's even drier, it'll be a much smoother process.
Yeah. All right, Joe, I realised you had a lot of questions. I do one more question for
Great.
Joe before we wrap up. This one's from Sandra Moran. Could you give any more detail on the market research you're doing for the biodiversity crediting approach? She's particularly interested how an approach which is sufficiently commercial maintains environmental integrity.
Yeah, so the market research that we're doing is a little bit more aimed at just getting an idea of what benefits outside of just a quantified biodiversity uplift do buyers and sellers want? Is it this standardized MRV, which we've heard from a lot of different landowners and project developers, so that they can compare across different sites or legitimize their biodiversity work by comparing it to an existing framework? From the buyer's side, it's what would give them market confidence? What would make them feel like their biodiversity credit that they're buying isn't going to be the face of a scandal 5 years from now? And because I work for the Peatland Code and Woodland Carbon Code, who do sell higher end carbon credits because of the integrity of those programs, we're going to lean more on the side of integrity versus a price point, basically, is the other side of it. It's not nice, but one feature of this market is that biodiversity credits don't really have a set selling price. And so right now it's going to be informed by how much it costs to do the monitoring and have an additional private finance pathway towards the restoration. So this second part of your statement to me doesn't necessarily seem like two things at odds. So often we see profit versus environmental integrity being pulled apart from each other. But because this market is being built from the ground up, basically the price of the credits will be informed by how much it costs to produce a high integrity credit, if that makes sense. So we don't have to try to force the price down, at least at this point in the market, because there's so much, because it's such a young market, there's potential for these different types of credits to pop up in different places. And quite frankly, not even every biodiversity crediting program agrees on what a biodiversity credit is. So of course, the integrity and even the meaning of these credits will be different between different creditors. One of the reasons we're aligning with Operation Wallacea is to bring some cohesion to this very complex market. But we don't really have to have that tension right now between commercial viability and environmental integrity. Of course, we want to make sure that it's not so cost prohibitive that it's unappealing to voluntary purchasers who want to do this for either ESG or showing that they're, representing a nature positive organization, or they want to have a positive contribution on ecosystem services that are important to their supply chain. But that's kind of how we're trying to deal with that tension between the market and environmental integrity.
Yeah, that's brilliant. And it's always been something that's really central to the Peatland Code. So it's great that's carrying on with our biodiversity work as well. Thank you, Joe. So that's all the questions that we've got time for today. But we will be sending out a link to the recording once it's ready on our YouTube channel. And Jessica's agreed to record her presentation as well. So, sadly you missed it today and we didn't get a chance to include Jessica in the Q&A, but you'll still have an opportunity to catch up on her presentation as well. We'll also send out a really brief questionnaire with that to get your feedback on today's webinar. I know we all get sent a million surveys, but we'd really appreciate you spending a few minutes to sort of share the topics you'd like to see us explore in future. We've got another webinar coming up as well at 2 p.m. on the 17th of April to celebrate the launch of our latest publication on forest to bog restoration. So we really hope you can join us for that as well. And you can find out more about that and sign up on the events pages of our website. The final plug from me as well is for our annual conference. So that's taking place from the 17th to the 19th of September this year in the beautiful Cairngorms of Scotland and online with the theme Peatlands, People and Nature. And we're excited to be releasing tickets for that next month. So keep your eyes peeled for that on our X and LinkedIn channels. And finally, I'd like to say a huge thank you to all of our speakers today for sharing their time and expertise in support of our webinar. It's been an absolutely fascinating explanation of so many different topics in one afternoon. And I really hope you've all enjoyed it, and do share that presentation with anyone else you think might be interested when it comes through. Thanks also to Claire for working busily away in the background and making sure things ran as smoothly as they could today. So brilliant. Thank you, everyone, and hope to see you again soon.